here.
In your opinion, can you give us the 5 most exciting things that you just spoke about? And the reason for that is that many of us have our database of donors. We would love to be able to go back right now to all the people that have donated to our walks and our snowmobile rides and our fishing tournaments and say, here's where all in the Ice Bucket Challenge, and say, here's where your money is gone and here's the exciting future. But we need that condensed so that we don't lose anybody.
So I suppose I put my 5 big things into 5 boxes for a reason. I think those 5 areas, so studies in drug discovery, we have 10 to 12 new ones which we plan on investing in, and we can send summaries and things once they're all finalized. Clinical phase 2 trial, very excited about the program. We will definitely have a few of those. They're 1.5 million each. It's already only a small amount we can invest, but we will have 2 to 3 of those hopefully. The IPS core and the Neuro Collaborative, because they are open source opportunities for everyone to have access to. And then genetics. I strongly believe if we understand all the genetics of ALS, we're a long way ahead. So fantastic work at New York Genome Center and Project Mine, and others that I can't really talk about but will be coming up. So I can help with our development team to prepare materials that are helpful to you.
You talked about the influx of, that came in from the Ice Bucket Challenge. Who's deciding how and where those dollars are being appropriated? And is your team part of that process in terms of saying, hey, let's take this chunk and put it toward, one of those 5 or others?
Okay, so the chunk that is dedicated to research is really something we spend with all, so I listed all those people at the end because they've been pivotally involved. Those are my colleagues all over the world with different expertise. We already had some structured programs, so the 4 big initiatives that were announced earlier were all strategic plans that had been in planning but lacked funds, so it was a natural opportunity, and they had this exquisite opportunity of really bringing in, as I said to you, understanding the clinical side, bringing in people with the disease, making them central to discovering ALS through genetics, through the omic studies, through stem cells. So that's why those were chosen. I've had advisors, and of course internally with our organization, we've had strategic discussions. And so for each of the programs, we have a drug development program, as I highlighted, which is a solicitation of ideas from all over. The clinical phase 2 is strategically important. So we have a strategy set out for the next 2 to 5 years, which actually is going to be in publication and rolled out very soon, which will give a little bit of breadth of that. In amongst that strategy, we have programs that have very specific calls for proposals. I was very passionate that as I said to you from the beginning, there are many ways you can spend your money. I don't think there's any right way or wrong way as long as it gets data out in the end. However, I was very passionate that we had, and I've been with the association for almost 15 years and built programs. I didn't want it only dictated by what me and my colleagues around me and a few others might think. I wanted to be sure that everyone had a chance to submit their ideas. At that point, because we have spent the Ice Bucket 5 times over with all the things that have come our way, we had to have some level of system where there are reviews and oversight, etc. And that was already very much in place because that's how we've built our programs over the years. Hopefully that helps a little bit. Yes, I see a hand.
Hi. I appreciate that the association, in terms of clinical trials, is really trying to put their resources towards the phase 2 trials, where a lot of the excitement and real challenges are. You talked about wanting to make sure we design the trials so we learn about what needs to go forward in terms of efficacy, but at the same time, when you talk about rigidibine, it's a small trial that's really powered for tolerability and for safety. And so I guess my question is, when we look at the literature, the biostat literature, there are many recommendations for how we get these 2 priorities to coalesce. But when we look at the types of trials Niels is recommending, it seems like we're heavily weighted towards the tolerability side, and we're left guessing in terms of which ones to move forward. If you could just talk for a minute about how we fix that system that people have been talking about for the past 5 years.
So your point that the trials are listed as phase 2 tolerability, because that, truthfully, for that size is what you can really do with a small size. You can ask about tolerability or dosing or things like that. It doesn't mean to say at the same time we're not building in measures to see if it affects something, and that's why the value of adding on the biomarker and these are shortish time frames, and some of the studies where a biomarker is very defined, it doesn't have to be a very long study. It doesn't have to be the phase 3 long survival trial because it's the only approved biomarker that might be relevant. These biomarker trials are quite short, and they then will become much more informative to be able to design the bigger study in a much quicker time frame to answer the question you have. So yes, they have to say tolerability, because it would be incorrect to say that those small size studies are looking for something efficacious. But by golly, if we get a good biomarker out of it, you can design a study much more effectively with probably smaller numbers of people, bigger than the phase 2, but smaller phase 3 trials that will very much dictate efficacy as well as things. So I think it might be perceived that they're all tolerability only, because that's realistically what you honestly can get out of it. But it is going to be a way of getting data to more quickly go to your efficacy trials. And also remember that we are an evolving clinical team over time. We've learned, and we've sometimes, as I said to you, because we are doing that now, we're forcing people to use biomarkers, it's not so easy. And the temptation, as we all know, is to try anything anyway, just in case it works. You know, one might say DEX at 80 million was a little bit of that experiment. We didn't have a way to measure if DEX engaged the target we thought was maybe mitochondria. We had actually no way of doing that. The company invested, and I'm not here to quote their reasons. We are grateful because that company is still with us, fantastic. But it was really an experiment that may have been doomed to fail. And was that hint in phase 2 really good enough? That's sort of the basis of how we have to think of changing things. I hope that helps a little bit. Yes. Oh, you need a microphone, sorry. And we have a microphone there, one moment.
Hi. I'm aware that some of these very exciting research things that you're talking about have been also undertaken by some other research groups, such as MGH has the biorepository, and ALS-TDI is doing the iPS stem cells and also mapping genomes. How much sharing is going on, and how much duplication of effort?
So first of all, the MGH biorepository is funded through our programs, and so it's very much under this umbrella. I didn't always exactly quote everyone, and it's just so complex. So the biorepository, which in fact we're about to announce an extension of that in Arizona, there'll be a second site for those. It's completely open source. Those are samples that are going to be accessible by researchers worldwide. One of the big things we've been pushing, and again, I failed to add in, we are working with all the teams to have a GUID, a global unique identifier, so that if a patient is in study X and study Y, they will carry with them that study tagged by that GUID. And so that can be then tracked, and you will be incorporated. If you've been in one study, we won't replicate that statistic, and we'll combine the studies. And we are actually also funding Neurobank, which is a huge databasing system that is tying all these different studies together so that we can logically, although there are many separate studies, analyze the data as one bigger study. As far as the iPS stem cell work is going, currently we don't have a direct collaboration with TDI. We're very open to discussing that. I think there is no harm in having multiple hubs of these things, because it's a huge volume of work that has to be done. It's a huge cost involved. I think the bottom line is coordinating these studies. And I do believe that the GUID and all the programs that we are funding and setting up, are open source in these particular areas of reagents we're getting from people. They are to be shared, they have to be shared, that's part of the contract, and they will be under a common identifier so that they will all be linked together. I hope that helps. Yes?
Can you hear me?
Yes, I can hear you.
Okay.
I'll repeat the question if it's a little quiet.
Okay. Can any patient be tested by the New York Genome Center?
So, no, can any patient be tested by the New York Genome Center? No, it's not a testing facility as such. It's part of studies. So, if you want to be part of a study, and we will be improving how we communicate all the studies going out because there's just so much going on at the moment, we will make sure. But the studies are linked in with teams of investigators. So, the New York Genome Center is responding to that study to do the sequencing and analysis. It's not a place where you would just knock on the door and say, I want to be tested. That said, once we have outlined clearly where all the studies are and the sites that are collecting and the centers, probably getting in through your clinical neurologist will be the way to go in.
Okay, I was curious. I have familial, and I have tested negative for C9 and SOD1. And to have my blood further tested was going to be over $10,000. So I have gotten in touch with the University of Miami and Northwestern. And Northwestern would really like to have my brain and my spinal cord when I do die. And I'm passionate about letting them have that. However, my question is, I can't find anyone who's willing to take it out of my body and send it to them. And I wondered if there was anyone you could put me in touch with.
Okay,
this is a big thing, apparently.
Yeah. So I'm surprised that Northwestern can't provide you that information because Dr. Sadiq is perhaps who you've been talking to,
Yes,
and
yes.
Nyla. I mean,
Right.
I think that they would be able to give you those clues. But if not, I know them well, and let's try and have a dialogue and see what we can do about that.
Thank you, yes. We've talked to the pathologist at Wake Forest, and as long as I don't croak on the weekend, we're good to go.
Yeah. Okay, so there shouldn't be I mean, I think that they should be able to work that out. So I'm not
Okay.
quite sure what it is that doesn't work at the moment, but I'll discuss it with you.
Thank you.
You raise a very good point, though, just before I go on to the next questions. With many of these studies, obviously as part of the study and trying to identify the genetics, it's not as if when you get into the study, you get a little list afterwards of the genes that are involved, because it might be much more complicated than that. We have to first prove, we will never be able to go back to you and say it's this gene X, Y, and Z before we're absolutely sure, because as I said to you, we have many changes individually anyway in our bodies. And so if we are going to say that it's exquisitely linked to your disease, we don't want to make an error and say, well, this is the gene. So you are part of a study, and this is obviously everything that will be discussed with you when you get into the study, but it's not as if it's a gene testing facility as part of that study. Just important to know. Yes.
In an ideal world of science or ALS science, you'll be looking at a target which a drug which targets both the upper and the lower motor neuron, the dendritic spine, the axons. Do you have any potential targets, any list of, potential candidates for this?
So that's a good, so your question is, what are the targets for the upper motor neuron system in particular?
The upper and the lower end.
Okay, so I think those are good questions, and I don't think there is a specific list of targets that are clear. I mean, obviously, there are the common death pathways that we think about that might affect upper and lower. But it's a good thing you mention it, because a lot of our work has often been on lower because it's easier to do those studies. But there are some great scientists, one in particular, Hande Oslander and Jeff Macklis, who are working at University of Chicago and MGH, focusing on the upper motor neuron and trying to see if they can fall out with targets. If you're particularly interested in that area, I can give you contacts to connect with them. But I don't have an easy list, unfortunately.
So do you believe, the next question is do you believe in dying forward hypothesis, or is it more a dying backward hypothesis?
Yeah, that's a good discussion, and I have to explain it. That could be a whole scientific debate. So the question really is, and I brought up the fact that the muscle and the nerve cells are key in the disease. I don't know if it's more an academic debate in my mind whether it's that way or not. So the question is, does disease start in the motor neuron, or does the disease start in the nerve cell? it's very hard to know in people living with the disease because the disease has already started at a much earlier time point. We have some interesting data from mouse models to say that maybe it originates in the muscle and then it's the nerve. But my argument is that maybe it's a combination because it might start there, but it might have been a cue from the nerve cell that caused the muscle to have that problem. It's a chicken and egg question, and I'm not absolutely sure that that fundamentally will change what we do. I think both ends are important, and figuring out how to treat both is critical. That's my view, not everyone's view.
Lucie, we're going to take 2 more questions.
Okay.
To remind everyone, we will be having the Meet the Research session coming up following the next presentation, so you'll have more time to ask questions to Lucie and other researchers. Following this, we will have a 30-minute refreshment break, but we will be starting the government research session promptly at 11 for everyone that's watching from home on the live stream. So we have 2 additional questions.
Hi, you had talked about that a treatment will most likely lie in the cocktail or combination drug approach. Do you think that really tech will be included in that combination?
So probably. I mean, I think most trials are, so it you know, in Europe, almost every person is on riluzole. It's just the way it is. So any European study, they already have riluzole. And for some, you know, it has a modest benefit. Others feel not. So the trials are always anyway arranged with on riluzole, not riluzole. So yes, until it's proven that it's not helpful, I think it is a reasonable thing to include. Again, it depends on tolerability of the person. If they feel that they really don't feel great on riluzole and it doesn't. But in terms of trial design, it will always be riluzole plus. At least certainly in the Europe, and often it is the case here. Last question.
Whatever happened to NP001?
So I can briefly say that we are looking at applications for doing a phase 2 trial on that. We haven't finalized review of it. NP001 showed a modest effect, not entirely statistically significant, depending on whether you'd used historical controls or not, so quite complicated. There was a hint of something. And what was particularly interesting is it seemed that they identified a population that would be more likely to respond than another population. So it fits very well as a phase 2 to develop the biomarkers. And so we are reviewing that to see if that's something to bring forward. Okay? I think that's